In today's lecture, we're going to keep discuss about the nucleic acid, especially focusing on its structure as well as on higher order structures too. Let's start with the primary structure of DNA, which is basically a linear 2D structure, consists of nucleotides. And as we've discussed in a previous lecture, the nucleotide is basically a pentose sugar with a phosphate group and nitrogenous base. And between these nucleotides, they can form the base pairing by the hydrogen bonding, and all the polymers of these nucleotides form double-stranded DNA. And importantly, they're always in a direction starting from the 5' to 3' for both strands. And the secondary structure of DNA is basically 3D structure of DNA double helix, as opposed to primary structure, again, was the array of nucleotides that was aligned from the 5' to 3' to show the sequence of those bases or the base pairs. This secondary structure of DNA shows the right-handed double helix, where you can wrap this double helix with your right hand with your fingers pointing upward. And this secondary structure is a highly stable structure maintained largely through the hydrogen bonding between the complementary bases on the different strands, and also the base stacking forces between the adjacent bases that was driven by the van der Waals forces. DNA can have mainly three different forms for its secondary structure, which are A, B, and Z forms. And among them, A and B forms of DNA have two non-equivalent groups, which are major and minor groups. But it rarely forms in these groups with the same sizes, especially under normal conditions. So these groups are basically the openings between successive turns of the sugar-phosphate backbones. And especially through these major groups, which exposes a few more base pairs, as you can see here, as opposed to minor group, is basically just a little chasm between these two major groups. The major groups and the base pairs in these major groups are exposed to the solvent, basically the water in the cells, to allow some kind of biochemical reactions with the soluble proteins. Regardless of the type or the location of the base pairs, they have the same geometry where their center for their turns are perfectly overlapped right in the middle of these base pairs, as well as in their distance to the sugar-phosphate backbones between two base paired nucleotides are identical as 10.85 angstrom. And here are some features of DNA groups which are important for biochemical reactions. First, each Watson-Crick base pair, such as GC, AT, CG, and TA, has a major group side and the minor group side, basically alternating these two type of groups. And on either side, especially on the major group, there is a potential for the base pairs to the hydrogen bonding within this nitrogenous base to the external groups such as water molecules or proteins for their biochemical reactions. And functional groups of base pairs can be assessed from the groups such as amino group, carbonyl group, or the nitrogen atom in the ring, and so on. And they serve as a contact point for the protein searching for specific sequences on the DNA. So some kind of proteins such as initiation factors for the DNA replication and so on, or the transcription factors and so on, can just sit on some of the DNA double helix and then slide down to find the specific regions that they're targeting. And in that case, they use these specific groups, functional groups, within the base pairs, rather than sugar phosphate, which is pretty much identical all along the double helix. And lastly, hydrophobic and van der Waals forces also act between the adjacent bases to contribute to the stability. There are three types of geometries for double-stranded helices. Two of them are the right-handed helices, which are B form and A form. And the B form is found in the cell And most of the DNA under normal conditions assume this structure, and it also fits well with the Watson-Crick model, as opposed to A form is a shorter and the broader helix, and the examples are the double-stranded RNA instead of DNA, and also RNA-DNA hybrids. And as you can see in this picture, the aDNA has the also minor and major groups, but their differences are less prominent than the bDNA form. And there's a left-handed helix too for the DNA, which is a Z form, as you can see here, turn is the other side, and can be formed with the particular sequences under special circumstances. Here's a comparison between different forms of DNA double helix structures. Like I told you in the previous slides, the bDNA is the standard structure, where it spans a 3.4 nanometer per turn, and it has 23.5 angstrom wide and also spans 10 base pair per turn. But the aDNA, which is a little bit squashed form, has a shorter nanometer per turn, and as a result, it has a wider diameter of the double helix as 25.5 angstrom. When you compare to bDNA, they're definitely wider, and also has one more base pair per turn too. And the zDNA, which is only shown in rare cases, in special cases, actually exhibits more like a stretched double helix structure, where it has a much longer distance, than, which is 4.5 nanometer per turn, and also has a shorter or smaller diameter, as you can see here when you compare it to the bDNA structure, and also spans up to more base pairs per turn. And when you look at these structures in the projection, where you see the top of the double helix, the bDNA locates all the base pairs right in the middle of this double helix. So they basically don't have any big openings in this double-stranded structure. But the aDNA now has the base pairs tilted about 19 degrees from the axis of the helix. As a result, these base pairs are a little pulled towards the backbone side, which leaves huge openings right in the middle of the double helix. And the zDNA is also have base pairs tilted about 9 degrees from the axis of the helix. And also, they pushes this base pairs a little bit from the center. Here are some more structural features of A, B, and Z DNA. Again, B is a standard, and you can basically compare that with the A DNA or the Z DNA in terms of their diameter, base pair per helical turns, or the helix rise per base pair, which has all varies, and also tilt, the angles, and how the major group and minor group look like compared to the B structure here, and also sugar puckers and so on, which we'll discuss a little bit later on. 6 torsion angles completely describe the conformation of a nucleotide in a polynucleotide chain, starting from alpha, go through the zeta, and also chi too. And there are several points that I want you to remember and keep in mind, especially for better understanding the structure of the DNA double helix, as well as how the base pairs are structured and located in there. First one is that the sugar phosphate torsion angles are very restricted, and they're not so free to rotate too. And you can easily imagine that because of the double helix of the DNA is quite stable, and also keep the same size for the major and minor groups and so on. And the base pairs are always pretty specific and located right in the middle of this double helix too. So they have not much room to rotate or move there. And the torsion angles or the rotates are largely depends on the requirement of imposed by the base pairing geometry itself, not about the backbones or the other molecules that might interact with the other proteins or the molecules. And the second point is that there are only 2 orientation of nitrogenous bases about N-glycosidic bonds are permissible. which are syn and anti. And syn is basically the form where the nitrogenous base with this bulky part is stacked on top of the pentose sugar. But the anti is this nitrogenous base that is turned 180 degree. So the little bulky part is moved away from the pentose sugar. And also pyrimidines can have the same kind of syn and anti form. The purines can have both the syn and anti because there's no steric clashes with the other atoms on the pentose sugar. But the pyrimidines such as cytosine, thymine, and uracil have this bulky oxygen atom that is located here with the two lone pairs of electrons that will have the steric clash with the C2' on the pentose sugar. So it will only have allow the antiform here, not the syn. And the third point is that when you look at the pentose sugar, the ring substituents are eclipsed if the sugar is completely flat, meaning that all these substituents are going to be too crowded and they're going to push each other or going to be on the way of other atoms. So that will cause some steric clashes. To avoid that, the sugar have a slight puckering, and there are two types called C2' and C3'. Basically, these 2' or 3' carbon atoms are pushed slightly out of the plane. So the C2' endo, which is one type of that puckering, can be observed typically on the bDNA, where the 2' carbon among the pentose sugar is pushed out of the plane of this pentose sugar, as opposed to C3' endo. Now the 3' atom is pushed and moved out of the plane, and it is shown in the aDNA. Here's a quick summary of the properties of the double-stranded DNA. First, two strands are held together by hydrogen bonds between the complementary base pairs, not any interactions between the phosphate or the backbones at all. And also there's a base pair stacking or van der Waals interactions between adjacent pairs, which also extra strength that stabilizes the double helix structure. And the strands run anti-parallel, meaning that when you look at it from the top of the double helix, one strand goes 5' to 3', but the other strand is looking on the top, starting from the 3' to the 5'. And both the formation and disruption of DNA double helices are highly cooperative, meaning that the one base pair disruption can lead to the other disruption of the base pairs in vicinity. And here's a table showing the stacking energies for the 10 possible dimers in bDNA, which ranges quite a bit from the AT to TA to GC to CG. DNA superhelicity is an interesting phenomenon where DNA forms helix over helix due to underlining of double-stranded DNA to basically packing down the DNA molecules within the cell. Then why does cell want to pack down the DNA molecules? And what does this DNA superhelicity do in terms of sustaining life? Let's compare some of these numbers between the size of the cells as well as its genome size. When you look at the E. coli bacterium, the size of it is about 1 micrometer or 10 to the 6th meter long, fairly small. But its genome size, the DNA molecules, is 1600 micrometer or 1.6 millimeter long, which is much larger than the size of the E. coli cell. The same thing applies to human cell. A human cell size is about 10 micrometer long, but its genome size is 1.8 micrometer or 1.8 meter long, which is much larger than the size of the human cell. So there's a necessity that cell has to deal with this really large or really long material that has to be contained within the cell and have to manage to do it for the replication as well as some kind of gene expression too. So in order to do that, cell has to come up with a way to contain, package, or handle their DNA. And some of the chromosomes are linear, For example, in eukaryotes and some viruses, the DNAs are linear, but some are circular too, such as bacteria and mitochondria, chloroplasts, and some viruses too. But in all cases, regardless of the linear or the circular shape of the chromosomes, the DNA has to be coiled very tightly in the cell or virus particles to basically to achieve this. The long, really long and large DNA molecules get in and fit it into the relatively small cell size. And relevant to superhelicity, a closed circular and double-stranded molecule of DNA can be compacted by a process called supercoiling, and this supercoiling results from overwinding or underwinding of DNA double helix prior to joining both ends of each strand. So it has to be joined, meaning that this DNA has to be circular or closed form again. And supercoiling can only exist in a molecule where both strands of DNA are closed circles. So if one breaks, then the DNA rapidly loses its supercoiling, so it relaxes, going back to this state. You can easily imagine that by having the rubber band, and if you twist it a lot, then it'll be coiled. But when you cut out one of the that coiled region of the rubber band, and then it will release really quickly, and then it looks like this relaxed form. And most closed circular DNA is supercoiled within the cells. To control the supercoiling and the superhelicity of the DNA molecules, the cells have topoisomerases, which are the enzymes that catalyze interconversion of topoisomers. And topoisomers are basically the same DNA molecules, but with the different twistings. And you can change the superhelicity. There are two different types of topoisomerases, topoisomerase 1 and topoisomerase 2. And topo 1 relaxes negatively supercoiled DNA, and this process can be done passively by nicking one strand, then the other. And a strand passes through the single strand break, and then the strand reseals in this process. But topoisomerase 2 has a slightly different mechanism for changing the superhelicity. It can relax or also introduce supercoils within this DNA molecules, and also it can create the high energy supercoils through ATP hydrolysis. So it's more like an active process rather than topoisomerase 1 that works passively. And lastly, topo 2 breaks both strands and then reseals both. Let's more talk about the eukaryotic chromosome structures. The length of DNA in the nucleus is far greater than the size of the compartment in which it is contained, as we discussed in earlier slides. And to fit into this compartment, the DNA has to be condensed in some manner, such as supercoiling. And the degree to which DNA is condensed is expressed as its packing ratio, where packing ratio is the length of DNA divided by the length into which it is packaged. So let's take a look at an example here. The shortest human chromosome contains 4.6 times 10 to the 7 base pair of DNA, which is about 10 times the greater of the genome size of E. coli. And this is equivalent to 14,000 micrometer of extended DNA, so it's the genome size. But in its most condensed state during mitosis, where the cells are divided, the chromosome is about 2 micrometer long, so it's much smaller or shorter than the actual extended DNA size. So that gives the packing ratio of 7,000, which is 14,000 here, divided by the 2, the shortest length here. Chromatin, which is a gather of DNA molecules, have a hierarchical organization, and its first level packing is nucleosome here, which is achieved by the winding of DNA around the protein core called histones to produce a bead-like structures, as you can see here, and the packing ratio is about 6. And this structure is invariant in both euchromatin and heterochromatin of all chromosomes. And as you know, euchromatin is the region of the chromatin where the gene expression is active, as opposed to heterochromatin is the region of the chromatin where the gene expression is inhibited. And the next level of packing is the 30 nanometer fiber, which is the second level of packing, and that is basically the coiling of beads, these little nucleosome beads, in a helical structure that is found in both interphase chromatin and mitotic chromosomes too, with a packing ratio about 40 or so. So it is highly packed here too. But there's a final packing too, which occurs when the fiber is organized in loops, as you can see here. And it forms has the scaffolds and the domains that give a final packing ratio of about 1,000 in interphase chromosomes, which is the cells not in the state of cell divisions, but it achieves the packing ratio of 1,000 in the mitotic chromosomes, which are the chromosomes in the cells that are dividing actively. Let's talk about the differences between chromosome and chromatins. Eukaryotic chromosomes consist of a DNA protein complex that is organized in a compact manner, and that permits the large amount of DNA to be stored in the nucleus of the cell, as we discussed in the last slide about packing the DNA. And the subunit designation of the chromosome is chromatin. The fundamental unit of chromatin is the nucleosome, which is the first level among the hierarchical organization of the chromosomes. And chromatin is the unit of analysis of the chromosome, and chromatin reflects the general structure of the chromosome, but it's not unique to any particular chromosomes. So here, what you're looking at is the chromosome, which is the whole highly packed DNA molecules during the mitotic phase. And each of this one chromosome consists of two sister chromatids, which is another term for calling the DNA molecules. And the nucleosome, which you can see in this electron micrograph as a black beads, is the simplest packaging structure of DNA that is found in all eukaryotic chromosomes. And DNA is wrapped around an octamer of small basic proteins called histones. And histones have subunits such as H2A, H2B, H3, and H4, and they put together to form the functional group as octamer here. And 146 base pairs of DNA is wrapped around the core protein of the histone on octamer here, and the remaining base is linked to the next chromosome or nucleosomes here and here too. And this structure causes negative supercoiling. Again, core histones within a nucleosome have two copies of histone protein subunits, H2A, H2B, H3, and H4, to form the octamer. And histones are basic proteins, so they have affinity for DNA, which is negatively charged due to its sugar phosphate backbone. And histones are the most abundant proteins associated with DNA, and also their amino acid sequences are highly conserved throughout all different life forms, suggesting a similar function for all those species, as well as the fact that all the residues are invariant, meaning that all the individual residues are critical for the function, and it won't allow any variation. The DNA that is associated with the histone octamer is called core DNA, and its size or the length is invariant as 146 base pairs, as we discussed earlier. The core DNA forms two loops around the octamer, as you can see here, and this permits two regions that are about 80 base pair apart to be brought into proximity. And thus, these two sequences, or the sequence regions that are far apart, can interact with the same regulatory protein to control the gene expression at the same time. And the DNA that is between each histone octamer is called the linker DNA, which is located here. and its size or the length can be vary from 8 to 114 base pairs, so it's highly variable. And this variation is species specific, but the variation in linker DNA length has also been associated with the developmental stages of the organism or specific regions of the genomes too. So it is relatively dynamic in terms of the regulating the size of the linkers. The second level of organization of the chromatin is the 30 nanometer fiber. And you can see it in the electron micrographs here too, which has a lot of beads get together, forming this kind of thick bundles. And this appears to be a solenoid structure, or sort of the helix of those little like nucleosome beads, with about 6 nucleosomes per turn. This gives a packing ratio of about 40, which means that every 1 micrometer along the axis of this 30 nanometer fiber, it contains the 40 micrometer of DNA. And the stability of this structure requires the presence of the last member of the histone gene family, which is H1, which we have never talked about yet, but it is located here between the linker and the nucleosome to stabilize this structure. But when there's an experiment to strip the H1 from the chromatin, it actually did not affect the nucleosome structure at all, but it only affected the 30 nanometer structure, which means that this H1 is important for the stabilization of the 30 nanometer structure. Here's a fancy 3D reconstruction structure of the 30 nanometer fiber, where you can see the nucleosomes here with the 2 turns of the DNA double helix, and also some of the histone subunits, and they make this kind of solenoid structure to form the 30 nanometer fiber. And the final level of packaging of DNA molecules is characterized by the 700 nanometer structure seen in the metaphase chromosomes. And the condensed piece of chromatin has a characteristic scaffolding structure, as you can see here, that can be detected in metaphase chromosomes. And this appears to be the result of an extensive looping everywhere of the DNA in chromosomes. And it can time to time have these loops too for certain gene expression too. So here's a recap for the different levels or hierarchical structure of the chromatin structures. It starts with the double-stranded DNA, which forms the nucleosome with the histones. And then these nucleosomes get together to form this solenoid structure, which is a 30 nanometer fiber. And these fibers are looped around to also packed into this even bigger, higher order structures to have to achieve the really high folding ratio. At the beginning of the last lecture, we talked about the central dogma of biology, that DNA makes RNA and RNA makes protein. But here's a little bit of a revision here and the addition as a special path too, where the DNA can actually make the protein directly in the lab using some bacterial systems such as E. coli to make the recombinant proteins too. And also, the RNA virus can actually generate DNA molecules out of their RNA genome, and also they can replicate themselves, and eventually they're going to make some proteins into the for their activities too. So let's talk much more about the RNAs from now on. RNA, the ribonucleic acid, is primarily made up of 4 different bases: adenine, guanine, cytosine, and uracil instead of thymine in DNA, but uracil still have the same base complementarity to adenine. And uracil is pyrimidine just like thymine, and it has a very similar structure to thymine too. However, uracil is energetically less expensive to produce than thymine, which may account for its use in RNA, which depends on the bulk production for gene expression, as well as a fast degradation for fast gene regulation. However, in DNA, uracil can also be readily produced by chemical degradation of cytosine, which is a bad thing. So having thymine as a normal base in DNA makes much more sense for detection and repair of such incipient mutations with a high efficiency. Therefore, uracil is more appropriate for RNA where the quantity is important. Again, RNA is for bulk and fast production for the fast gene expression as well as a rapid degradation for stopping that expression at certain conditions. But lifespan is not really for the RNA or the uracil usage, whereas thymine is appropriate for DNA where maintaining sequence with a high fidelity is more critical. And here it shows the nucleotide structure differences between DNA and RNA, where the DNA uses deoxyribose, RNA uses the ribose. And we talked about this, that RNA has the hydroxyl group instead of hydrogen atom at C2'. And this is the examples of the bases that is only specific to this type of nucleotides, which is the DNA is for thymine, and the RNA uses the uracil. Have a really similar structure except for this one hydrogen atom. Unlike DNA, RNA is often a single-stranded molecule and has a much shorter chain of nucleotides. Again, it sacrifices some of the stability as well as length too, but it's only for the expression of certain genes to produce the protein, that particular responding proteins. And RNA, this is what we talked already about, different types of pentose, and also the hydroxyl group that has different positions. And this hydroxyl group makes the RNA less stable than DNA because it is more prone to hydrolysis. A second consequence of the presence of the 2' carbon hydroxyl group is that it enforces the C3' endo sugar conformation as opposed to the C2' endo conformation of the deoxyribose sugar in DNA. And this causes the helix to adopt the A form geometry rather than the B form that is most commonly observed in DNA. And this results in a very deep, narrow major group and a shallow and wide minor groups that we discussed earlier in this lecture. And single-stranded RNA exhibits a right-handed stacking pattern that is stabilized by base stacking too, which is enforced by the van der Waals force. There are also numerous modified bases and sugars found in RNA that serve many different roles. For example, surouridine, which can be written as Psi, and the DNA nucleoside T are found in various places. For example, T-Psi-C loop of every transfer RNA. And another notable modified base is inosine, which is a deaminated guanine base that allows a wobble codon sequence in tRNA, which gives a flexibility in terms of this tRNA coding some of the amino acids. And there are nearly 100 other naturally occurring modified bases, of which pseudouridine and the 2'-O methylribose are by far the most common ones, and they have a strong and really important functions in terms of the biochemical reactions and their regulations that we're going to talk about it much later in this course. And this specific roles of many of these modifications in RNA are not fully understood, and still an interesting field that many of the researchers are pursuing. And all the functions that is known so far is this modification in RNA regulate the RNA stability, localization, translation patterns, as well as responding to some kind of environmental cues and so on. And in ribosomal RNA, many of the post-transcriptional modifications occur in highly functional groups such as peptidotransferase center of the ribosomal RNA or the subunit interface of ribosomes, which is inferring that there are important, they are really important for the normal functions. And also, if you get rid of some of these modifications, you can actually see that this ribosomal RNA or the ribosome doesn't function properly. Like DNA, most biologically active RNAs, including transfer RNA, ribosomal RNA, and small nuclear RNAs, and other non-coding RNAs too, are extensively base paired to form double-stranded helices, as well as interaction between the RNA and DNA too. And structural analysis of these RNAs have revealed that they are not single-stranded, but rather highly structured to form certain kind of hairpin-like shapes or some other type of secondary structures. Unlike DNA, though, this structure is not just limited to long double-stranded helices, but rather collections of short helices here and there, packed together into a structure of some kind of blob or the globular structure that is similar to proteins. And in this fashion, RNA can achieve chemical catalysis too, working something similar to the proteins too in terms of their functionality, like enzymes. For instances, the determination of the structure of the ribosome revealed that the active site of this enzyme that catalyzes the peptide bond formation is composed of entirely of RNA, meaning that this enzyme is actually dependent on the RNA and its RNA's function for forming this peptide bond. And several types of RNAs, including transfer RNAs and ribosomal RNAs, contain a great deal of secondary structure, like we talked about, sort of the globular or the blob structure, which help promote stability as well as the function of the RNA molecules too. And RNA secondary structure is generally divided into helices, which is by the contiguous base pairs, and the various kinds of loops too, which is done by the unpaired nucleotides surrounded by helices. So you can see the loops here, internal loops, and there could be some other loops too, and also some of the helices that is formed by the base pairing within the one RNA molecule. And this is an example of a secondary and tertiary structure of 5S RNA, but as you can see, the actual tertiary structure looks a little bit different than this secondary structure, because not only they make these loops and helices, they also have some interactions, the base pair interactions between some residues far away from the other to form these particular shapes. The RNA world hypothesis proposes that the earliest form of life relies on RNA rather than DNA or protein, because RNA can carry the genetic information too, just like DNA does, and also RNA can catalyze the biochemical reactions just like enzymes too. So according to this RNA world hypothesis, descendants of these early life forms gradually integrated DNA and proteins into their metabolism, but they started with the old RNAs.